Hi, I'm Mindy, and this is my research proposal, which aims to analyze the correlation between sunlight and the amount of algae. So, throughout this presentation, we will be discussing if there is a relationship between sunlight amount and the amount of algae. Are these factors connected, or are these factors independent from one another? I devised a few hypotheses to explain the data shown later in this presentation. The first is my null hypothesis, which states that there is no correlation between algal biomass and sunlight levels. If this null hypothesis is correct, then that means that there is no significant difference between the two factors. My other two hypotheses evaluates if there is any increase in algal biomass with sunnier or shadier regions. So, what exactly is algae and biomass? Algae is a diverse group of photosynthetic organisms that exist in almost all aquatic ecosystems. Collectively, photosynthetic plants like algae provide the food needed to sustain life in the ecosystem. To do so, they utilize the same source of energy, sunlight. Other animals are then able to consume these plants and move the obtained energy up the food chain. If the weight of all living organisms in an area is combined, it is called the biomass. Biomass differs due to a multitude of factors such as exposure to sunlight and water availability. The focus of this experiment, though, is based on a single factor, sunlight exposure, to understand its effect on the quantity of biomass. Biomass is an important measure in all ecosystems because it reflects the sunlight, water, and mineral levels a plant can harvest to turn into plant mass. Biomass cannot be measured directly because the presence of water does not account for plant mass. That's why we use ash-free dry mass, a way to measure biomass without the water by measuring carbon biomass, done by measuring the mass of the dried sample after it has been incinerated. The purpose of the incineration is to evaporate any available water remaining in the sample. This experiment was done in 3 portions: algae harvesting, algae sample filtration, and ash-free dry mass determination. The purpose of Part A is to collect the algae needed to sample for the AFDM. In this part, we collected the colonized portion of a cobble. My cobble especially was very dense with algae, resulting in thick and dark green liquid. In Part B, the vacuum manifold, which is shown on the next slide, is set up to collect the sample. For AFDM analysis, the mass of one piece of pre-combusted glass fiber filter paper is recorded. The filter paper was placed into the vacuum manifold to pump out most of the water from the sample, leaving behind the algae. This can only be done by measuring out 1 microliter increments of the sample, or else it will clog. When the filter is saturated with algae and the vacuum can no longer pump out water, remove and dispose of the waste accordingly. In Part C, the now saturated filter paper is baked in an oven to evaporate any water that may be in the sample. This is the crucial part of the AFDM measurement. After a day, move the algae to a furnace and allow it to cool. Return the next day and weigh the sample. This is now purely oxidized algae. Subtract the mass of the filter paper initially recorded to obtain the true mass of the algae biomass. These are the instruments that was used for the filter manifold, and the diagram on the right shows how the vacuum was set up. This experiment was carried out in 3 different general locations. Within each general location, samples were collected in both the sunny and shady regions for a total of 6 regions. This data is just to show the percent of canopy cover for the shady regions is higher than the percent of canopy cover for the sunny regions. The difference in distance between the sunny and shady regions should be no more than a couple of kilometers apart. For this experiment, the names of the locations do not have to be known. According to the data, the AFDM measurements are higher for the shadier areas than sunnier areas, besides the exception of location 3, where the average AFDM of the sunny region is slightly higher than the average AFDM of the shady region. Besides that slight difference between the 2 variables of location 3, there is a significant difference between the sunny and shady regions of location 1 and 2. The This is evidence that there is more biomass in shady locations than sunny locations. The data may not represent the exact biomass of the area because although most of the algae were collected from the cobbles, there was still residue on the rock, the scraping tools, and the collection tray. The data was also taken from different students, so any errors remain unknown and precision varies from cobble to cobble. The p-test calculation taken from the AFDM data is 0.585. Because the p-value is less than the a value of 3.499, we cannot reject the null hypothesis. Because the null hypothesis cannot be rejected, there is no statistical significance, and the conclusion is that there is no correlation between the amount of sunlight and the amount of biomass at AFDM. It is statistically insignificant because the data collected provide little evidence that the null hypothesis is false. The data collected is expected and unsurprising because algae can actually grow more efficiently when there is an alternation of light and dark periods. It stimulates photosynthetic efficiency because dark cycles allow for deoxidation of the electron transporters. Furthermore, the lack of alteration can lead to radiation damage to the algae. Thus, the hypothesis that biomass will increase with more sunlight can be rejected. The data will require more experimentation as the findings do not align and provide a definite conclusion. Conducting multiple trials in the same area could clarify the findings because the particulate matter in pollution can add to the AFDM mass. Pollution can vary from location to location. According to the Autotrophic Index, location 3 is highly polluted and could have an effect on the AFDM results because it adds particulate matter to the biomass measurements. On the other hand, locations 1 and 2 have low autotrophic indexes, below what is considered to be polluted. Using a single location multiple times would be more accurate. This experiment is important to understand the measure of productivity in the area because more productivity is a direct correlation to diverse species and polyculture. Thus, preventative measures can be taken to make sure that adequate biomass levels exist all throughout Waller Creek for the life that it should support, not just certain parts of the creek.